Good evening, everyone. I know that this is a little bit delayed from whenever we actually covered this in lecture. We covered this material I'm about to talk about on Monday. I guess that would be the 4th. Yeah, Monday the 4th. Well, what we're going to talk about today is nucleic acids. We're going to be talking about differences between them as well as the nitrogenous base pairs that make them up and everything like that. So stay tuned. All right, so the first thing that I want to go ahead and get started on is some definitions. One very important definition is the difference between a nucleotide and a nucleoside. This difference is pretty key to, well, just sort of using the language whenever you're talking about these two different things. A nucleoside consists of a nitrogenous base and plus a sugar. A nucleotide, on the other hand, is, well, both of those things, a nitrogenous base and a sugar, but has also at least one phosphate group. So these are pretty significant, from a structural standpoint, but also kind of a nomenclature standpoint. One of the terms that you'll hear interchangeably is a nucleotide triphosphate or a nucleoside triphosphate. And in both cases, you are, well, describing a nitrogenous base attached to a sugar with a phosphate group. But it's important to know and be able to discern those differences. Now, whenever we look at these two different things, one of the most important parts about this is the nitrogenous base. Nitrogenous base versus the sugar, we have to start somewhere. We're going to go ahead and start with our nitrogenous bases. Now, those bases fall into two different categories: purines and pyrimidines. Now, sorry about that, pyrimidines. Now, those purines and pyrimidines, well, they have kind of two different, or they have structurally different characteristics. For instance, a purine, and I'm going to go ahead and cross out this 2. What I was talking about there was the two different groups, purines versus pyrimidines. The purines, well, they have a general structure of a 6-membered ring plus a 5-membered ring. So it's a 2-ring structure that looks roughly like this. Whereas a pyrimidine is just a 6-membered ring. 6 members, or 6-membered ring, which just looks a little bit like this. Oopsies. Can I undo that one? Let's do that again. Looks a little bit like this. Now, where do these attach to the sugar? Well, in the case of your purines, you're going to attach down here, and a pyrimidine, you're going to attach down here. Okay. Now, ultimately, though, you want to know the two different groups and overall the structure of those molecules. So these these purines and pyrimidines, they ultimately break down into two different, or a couple of different structures. For instance, the purines, if you are talking about a DNA molecule, the purines that you are going to be talking about are adenosine and guanine. Now, if you are talking about RNA, you're talking about the exact same molecules. Pyrimidines, on the other hand, things are a little bit different. If you're talking about DNA, a DNA molecule, the nitrogenous bases that you're going to be talking about are cytosine and thymine. If you are talking about RNA, on the other hand, ribonucleic acid, you are talking about different molecules. You're talking about cytosine, that's consistent, cytosine, and uracil. Okay, now each of these molecules has a single letter code to recognize them. Adenosine has the letter A, guanine has the letter G, cytosine, the letter C, uracil, the letter U, cytosine again has the letter C, and thymine has the letter T. Now it's quite common to see something like a DNA sequence that is described as A, T, G, C, A, C, C, and so on. What's significant about this is that anytime you're talking about a double-stranded DNA molecule or a dsDNA, it's implied that this sequence that you've listed is labeled, this is referred to as the primary sequence, and it is read 5' to 3'. And with that, we have a reverse complementary molecule that, well, basically just matches that up. So we would label it from the 5' to 3' end. So C pairs up with G, G, T, G, C, A, T. Okay. So on the next, I'm going to go ahead and proceed on from here, and we're going to start talking about actually drawing these molecules. You're not going to have to draw the molecules, but it's important to know what they look like and be able to quickly identify them. Now, I'm going to go ahead and start out by drawing a molecule of adenosine. And whenever I do that, I'm going to label or number off all of the different molecules within that two-ring structure. Okay, so adenosine looks like such. And I'm going to start from the end where we are fixing it to our sugar. So we've got this 1, 2, 3, 4, 5-membered ring. Now, adjacent to that is our 6-membered ring. And what does that have? N, N, there we go. Okay, so we've got a double bond right here. We've got a double bond here, and here, and here. Okay, now this is going to be a resonance stabilized molecule. We want to go ahead and put on here NH2. Then let's see what else. That is actually everything. So I'm going to switch different colors to show a couple of things. There, this is our sugar from there. Now I want to switch to an even different color to go ahead and do the numbering. So the numbering of this molecule, numbering of the atoms within this molecule. 1, 2, 3, 4, 5, 6. So 6 to complete our ring. That's 7, 8, 9. So it's atom number 9 that attaches to your sugar. Now what's very important to recognize about this is just numbering 1 through 9. There's no other qualifier or anything like that. You don't say 1A or anything like that when you're talking about numbering the atoms within a sugar. Or, sorry, within a nitrogenous base. Now I'm going to draw our other purine, guanine. The same overall structure. There are some differences though. And in this case, I'm going to start with my 6-membered ring. So N, N, and there we go. We got a double bonded oxygen right there. I probably should have moved this over, but oh well, too late now. Then N, boom, boom, N, boom. Okay, I want to go ahead and fill in my double bonds here. There's one there, there's one there, and that would be it for this guy. Therefore, this guy has to have a hydrogen. In that position, we've got H2N. Okay, so now, again, I'm going to switch to blue to show where the sugar hooks up with it. Now switching to red to do the numbering of the atoms within this molecule. 1, 2, 3, 4, 5, 6, 7, 8, 9. Again, it is atom number 9 within this molecule that attaches to our sugar. And I'm going to go ahead and write sugar again right here. Okay, next up are our pyrimidines, which our pyrimidines are thymine and cytosine. And I'm going to switch to a different color just so you know that this molecule is a little bit of an outlier in that it is affiliated with RNA, and that is uracil. Okay, so thymine has the following structure. Nitrogen, we got double bond. NH, oxygen, a methyl group right here, and then that will do it. Okay, now for our numbering, I'll go into red, and I got a label down here, 1, 2, 3, 4, 5, 6. Now for cytosine, I'm going to go with the same color, nitrogen at the bottom. Oh yeah, and again, sugar. Sugar. Okay. Oh, sorry about that. Let me correct that guy. Okay, there we go. There we go. Nitrogen. Oh, that's supposed to be closed in there. Double bonds here, here, and then at the 2 position we have that double bond on oxygen, and then at the 4 position H2N, and there you go. Okay, numbering all our atoms here, 1, 2, 3, 4, 5, 6. And then uracil, our last molecule here, I'm going to go ahead and draw this with basically the exact same format. Nitrogen down there. We've got, there we go. We have, after that, oh, there's our nitrogen. So we have 2 nitrogens within the ring, just as we do with all the other molecules. There we go, and here we go, and I believe that would do it. So the difference between, I think one very effective way to study these molecules and be familiar with them is to simply look at what makes them different. If you look at uracil and thymine, they're really quite similar. The only difference is the methylation at position number 3, 4, 5. So within the ring of uracil, you've got a carbon, you've got a carbon at position number 5, and on uracil, well, it's bound to nothing, and in thymine, you have a methyl group. So I think it's important to recognize those differences because that will help you when it comes to actually knowing what that molecule is. Okay, so now what I want to do is I want to just go ahead and draw a molecule of ATP, adenosine, I forgot the A, adenosine triphosphate. So this is our nitrogenous base hooked up to a sugar, and we're going to go ahead and draw this for DNA. Adenosine triphosphate. We're going to go ahead and draw what this actually looks like, and we're going to start with our adenosine. So the first thing is N, then after that we've got to complete our ring. Okay, so adenosine, we've got those 2 nitrogens with our 6-membered ring. We have a double bond right here. We have a double bond right here, we have a double bond right here, and we have a double bond right here. The important thing is position number 6, we've got an NH2. Okay, now we can go ahead and connect this to our sugar. Okay, our sugar in this case, when we're talking about a DNA molecule, is deoxyribose. And this is actually a sugar where we do not have an oxygen at a specific position. Now, what we want to do there is we're going to go ahead and draw our sugar right here. Oxygen. Okay, and then whenever we do that, one of the important parts about all of this is to know what connects to what. So we have so here, this 5-membered ring right here, I'm going to put a little star by it. That is our sugar. Okay. Now, when you see something like deoxyribose, a sugar for a DNA molecule, this position right here is very important. This is the 2nd member of the ring. And this happens to be known as the 2' position. Because if you remember when we were talking about our nitrogenous bases, we numbered them 1 through 9. Or we numbered the atoms within those rings 1 through 9. We didn't call them 1', 1A, anything like that. So in order to kind of continue with that trend, or to actually avoid continuing with that trend, what we do is we identify the atoms within our sugar as a 1', 2', 3', 4', 5'. Okay, and so when we're talking about something like deoxyribose, we have a hydrogen right here. We do not have that hydroxyl group, but instead we have a hydrogen right there. Whereas at our 3' position, we have a hydroxyl group. Okay, now in order to actually connect this to our phosphate groups, in order for it to be an adenosine triphosphate molecule, what we have to have is carbon, or position 5' on our sugar has to have the hydroxyl group that is then ultimately phosphorylated and makes this chain of phosphate molecules. O, P, O, 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 and then double bonds, O, O, O. Okay, we have to put negative charges in there. 1, 2, 3, 4. Now, this is an adenosine triphosphate molecule. This is also known as D-ATP. You add in that D there because of the fact that this is a deoxyribose sugar. Now, in the event that we had a hydroxyl group there, well, we would be making an ATP molecule. So I'm just going to go ahead and add that in there. We would be making an ATP molecule, which would mean that it's really more effectively utilized for something, or synthesis of RNA rather than DNA. Okay, so we've made a DATP molecule. One other thing that I want you to be familiar with for this is the nomenclature associated with these phosphates. So this first phosphate off of our 5' carbon, this is going to be referred to as the alpha. The next one is the beta, and the third and final one is your gamma phosphate group. Okay, now this is significant because ATP is a high energy molecule. It's produced in the mitochondria. It's going to be used over and over again as you continue on into a metabolism class or something like that. At any rate, I wanted to show you this, and I also wanted to mention to you that just as we have ATP, which indicates there is no deoxy, instead this would be like a 2' and 3', both have OH groups. ATP, only 3' has OH group. There is also DDATP. This is dideoxy adenosine triphosphate. There are no OH groups at the 2' or the 3' position. I wanted to show all you this. I hope that you got some value from this. I would work through being able to recognize the multiple different types of the multiple different nitrogenous bases. We didn't spend a whole lot of time on sugars here, and that's just because they're kind of straightforward. The only major difference is the presence of the hydroxyl group on the 2' or 3' position. I would review this. I would also make sure that I know the difference between the nucleoside, nucleotide, and everything like that. But anyway, I hope this is valuable for y'all. Have a good evening. Thank you very much for watching.